Hello, my name is Dr. Jonny Furze and I'm a chemist at the University of Bristol. I'm going to give you a short introduction into ultraviolet visible light spectroscopy, or UV-Vis spectroscopy, and explain and then demonstrate how a UV-Vis machine works. So what is UV-Vis spectroscopy? Spectroscopy is the study of how the electromagnetic spectrum, or light of different wavelengths and energies, interacts with matter. UV-Vis spectroscopy specifically uses a range of light in the ultraviolet wavelengths and the full range of visible wavelengths. UV-Vis is one of a large selection of analytical techniques at a chemist's disposal: NMR spectroscopy, mass spectrometry, and X-ray spectroscopy, to name a few others. And by combining these techniques, you can learn a lot about a compound. For example, many chemists use UV-Vis spectroscopy on a daily basis to help calculate the concentration of a compound within a solution. The electromagnetic spectrum describes the different photonic energies of light and the related wavelengths or frequencies. The longer the wavelength of light, moving towards the radio side or the left-hand side of the spectrum, the shorter the frequency and the lower the energy each related photon has. The reverse is also true. The shorter the wavelength of light, moving towards the gamma ray side or the right-hand side of the spectrum, the higher the frequency and the higher the energy each related photon has. Due to their properties, different sections of the spectrum are able to interact with matter in different ways. For example, higher frequency microwaves are absorbed by molecules in food, increasing the molecule's internal rotational energy and therefore heating up the food. X-rays can pass easily through soft tissues such as muscle or fat cells, but get absorbed by bone, hence why the bones show up when you have an X-ray scan of a human body. Visible light is part of the spectrum that interacts with our eyes in a way that we see as color. For this talk, we're focusing on the ultraviolet and visible sections of the electromagnetic spectrum and how these interact with matter, hence the term UV-Vis spectroscopy. The UV range extends from 100 to 400 nanometers, and the visible range extends from 400 to 750 nanometers. So what does UV-Vis light interact with? UV-Vis light interacts with specific electrons within a molecule. For example, the delocalized electrons that are part of the C-to-C double bond found between two carbon atoms, or those found within transition metal compounds. The ultraviolet or visible wavelengths are of the correct wavelength to be absorbed and to give these electrons energy, exciting the electrons into specific higher energy states. Depending on the two atoms involved in the bond, the number of double bonds that are conjugated, or the transition metal complexes that are involved, will determine which section of the spectrum that is absorbed. A classic example of how UV-Vis light interacts with matter is how it interacts with plants, or more specifically, the molecules within the leaf that are associated with photosynthesis. Here, we show a UV-Vis spectrum with light wavelengths in nanometers along the x-axis and absorbance, which is unitless, along the y-axis. A UV-Vis spectrum displays a wavelength range generally from 300 nanometers, which is not detectable by the human eye, to 750 nanometers, which is seen as red light. The wavelengths that are absorbed by the sample are seen as peaks in the spectrum, and the wavelengths that are transmitted or reflected by the sample, and therefore the light that is detected by the detector, are seen as blank space in the spectrum. Chlorophyll A and chlorophyll B, shown here as the solid line and the dashed line respectively, both absorb light in the UV or blue and orange or red ranges of the spectrum, displayed on the spectrum here as large peaks in these regions. This means that the green or yellow areas of the spectrum are not absorbed and are reflected, being detected by our eyes. Why is ultraviolet or visible light absorbed by molecules? When a double bond is formed, the s atomic orbitals from each atom in the bond combine to form a sigma molecule bonding orbital, and the p orbitals from each atom in the bond combine to form pi molecular bonding orbitals. Rather than being associated with one particular atom, as they were when in the atomic orbitals, the electrons in the pi molecular bond become delocalized meaning they sit somewhere between the two atoms that are involved in the bond, both above and below the sigma bond, as shown by the yellow croissant shapes here in the image. Delocalized pi electrons in the double bond can absorb bond-specific wavelengths of light in the UV-Vis region, exciting the electrons. For example, a radio wave doesn't have a high enough energy to excite the electrons to a higher energy state available, but a UV wave does. When one double bond is formed, you get delocalized electrons in the pi molecular bonding orbital of that bond. However, if you have conjugated bonds in your molecule, the p orbitals of all the neighboring atoms making up the conjugated group combine and form a collection of delocalized electrons across the molecule. This has the effect of changing the wavelength of light that gets absorbed because the available energy levels have changed due to the conjugation. For example, beta-carotene, shown here, has 11 conjugated bonds, highlighted in red, and is seen as a red-orange colored molecule. The color comes about due to the conjugated system absorbing lots of the blue end of the spectrum, reflecting the rest of the visible light, which gives it the strong orange color. Going back to the example of plants and how UV-Vis light interacts with a leaf, you can see that the molecules associated with photosynthesis, chlorophyll A and B, have incredibly complex structures with many different conjugated bonds, creating chromophores, or areas where UV or visible light can be absorbed. These molecules absorb wavelengths in the UV and red ends of the spectrum, as shown on the spectrum here, reflecting the greener wavelengths, giving them a green color when observed through the human eye. Leaves of different plants have different amounts or concentrations of these chromophores within their leaves, as well as additional light-absorbing molecules, which is why some leaves are different shades of green, or even different colors, have different amounts and wavelengths of light are reflected and detected by our eyes. If you know that your molecule has a strong absorbance in the 200 to 800 nanometer wavelength range, then you can detect your molecule using UV-Vis spectroscopy. Each molecule will have a specific range of wavelengths that it will absorb, as mentioned earlier, but there will be one wavelength out of the range that is absorbed the most strongly, and it is this wavelength that is used for the molecule's detection. It's called the maximum absorbance wavelength. In the example shown in the image, the red and orange wavelengths are absorbed by the molecule, and therefore wouldn't be detected by a light-sensitive detector. If there is a higher concentration of the molecule in the solution, shown above, more red or orange light will be absorbed and therefore less transmitted and detected, making the maximum absorbance higher. You can use this correlation between concentration and absorbance as a method to work out the concentration of such a molecule through the creation of a calibration curve. Absorption of light in a sample that contains a light-absorbing molecule is determined by the molecule or molecules that are absorbing the light, and also how much of that molecule is in between the light source and the detector, or the concentration of the molecule in the solution. The Beer-Lambert law, shown here, is a way of describing this relationship. Light absorbed, A, relates to the molar attenuation coefficient of the sample, epsilon, to the depth of the sample, or the path length of the sample that the light has to pass through, L, and the molar concentration of the sample, C. The molar attenuation coefficient is a property that every molecule has and refers to how strongly a molecule reduces the intensity of light at a specific wavelength. Usually the maximum absorbance wavelength is used, and therefore the law changes slightly. The law states that if you double the concentration, then you double the absorbance. This also means that if you halve the concentration, you halve the absorbance. If the wavelength, and therefore the molar attenuation coefficient, and the path length remain constant, then a change in concentration will change the absorbance by a relative amount. By using this UV-Vis technique alongside the Beer-Lambert law, you can work out the concentration of a UV-Vis light absorbing molecule through the creation of a calibration curve. If a known concentration of sample is measured, this creates a known data point on a concentration absorbance graph. By plotting at least 3, but generally more, different known concentrations, a calibration curve can be generated. Now, if you have an unknown concentration of sample, the absorbance of the sample at the maximum absorbance wavelength can be plotted on the graph, and the concentration can be determined by drawing a straight line from the absorbance on the y-axis to the calibration curve, and then another straight line from the calibration curve to the x-axis. Here is a simplistic view of how a UV-Vis spectrometer functions. Light is emitted from a light source and is split by a prism to spread the wavelengths of light out. This light is then passed through the sample, with all the light that is not being absorbed by the sample getting detected by the detector. The image on the right shows you a slightly more complex view of the internal layout of a specific UV-Vis spectrometer, with adjustable mirrors giving control over which wavelengths of light are being passed through the sample over time.
The following video shows you how to set up the UV-Vis machine, setting the desired wavelength range and the running of a blank sample before any other samples are run. It's important to run a blank sample before taking sample measurements, as molecules in the air and within your solvent can absorb the analyzing light, giving you a false reading. It's also key to have clean sides to your sample holder, known as a cuvette. If you've handled the cuvette by the faces with your bare skin, oils will transfer to the surface and be detected by the machine, again giving you a false reading. The blank is run using the cuvette filled with your solvent. A solvent is the main liquid phase that has dissolved the substance or the solute you are analyzing. For example, in a sugar-water solution, the water is the solvent and the sugar is the solute. This is an image of a clean and empty plastic cuvette. The far UV wavelengths of light are absorbed by the plastic of this cuvette, therefore a quartz cuvette would be required if analyzing this area of the spectrum. The following video shows you how to flush through your cuvette. You want to clean out the cuvette with distilled water prior to flushing. Flushing the cuvette involves filling it with the sample that you are about to analyze and then disposing of this wash. This prevents any droplets of water used for cleaning remaining in the cuvette. These droplets would change the concentration of your sample and give you an incorrect reading. Once flushed, fill the cuvette roughly 3/4 full, ensuring that the area of the cuvette that the analyzing light passes through is filled with sample. Also check that the faces are dry, as even a small droplet of sample would change the path length and give you a false absorbance reading.
In the following video, we show how to run 3 different concentrations of a yellow solution, which is a yellow food dye dissolved in water. Food dyes are generally complex conjugated molecules and therefore absorb strongly in the UV-Vis range. Firstly, we remove the blank and place the first sample cuvette into the machine. The machine shines the selected range of light through the sample, 300 to 800 nanometers, and displays the results once finished. The maximum absorbance wavelength of the yellow food dye is 418 nanometers, so the wavelength is set to this. The concentration of the second sample is 3 times that of the first, seen in the absorption value being roughly 0.34 relative to the first sample, that was 0.11. The concentration of the final sample is 2 times that of the second, seen in the absorption value of 0.7 relative to the 0.34 of the second sample. When all samples are compared to each other, you can see a clear characteristic spectrum for the yellow dye molecules, absorbing strongly in the 380 to 480 region, with a peak absorption being at 418 nanometers. We ran 2 other food dyes. Each food dye is a differently conjugated molecule that absorbs strongly in certain areas of the UV-Vis spectrum, and transmits the other wavelengths, giving the molecule their color. The molecule giving the blue color absorbs strongly at the 620 to 850 range, which is the top spectrum. The molecule giving the yellow color absorbs strongly in the 380 to 480 nanometer range. This is the middle spectrum. The molecule giving a red color absorbs strongly in the 460 to 520 nanometer range. This is the bottom spectrum. So in summary, UV-Vis spectroscopy is an analytical technique that utilizes the absorption of a UV-Vis wavelength photon within a molecule to study a sample. The absorption of these photons excites an electron within the molecule that is part of a conjugated system, meaning that this wavelength of light is not transmitted through the sample. By using Beer-Lambert's law and a calibration curve that is specific to a certain molecule, you can use UV-Vis spectroscopy to calculate the concentration of a sample that is of unknown concentration. And finally, UV-Vis spectrometer is a very simple machine to use. Thank you very much for listening, and if you'd like to view more chemistry videos, please search for Bristol Chem Labs on YouTube. Have a great day.